Are you tired of doing cell culture in your kitchen?
Contaminated again? Ugh. I hate doing cell culture in a baking pan.
It's time for that new. Alright, here's a tool I made mostly for Suzanne because it's her protocol, but anybody else is using the same protocol. This is an app that'll do all the math. I think it's all the math needed in PBMC, cell plating, and then drug treatment. So this goes with the couple of Google Docs that are linked here that go through the math by hand that we would usually do and triple check, quadruple check with a bunch of paranoia, both in plating and then in treating with drugs. And those are linked in the spreadsheet, and then they also link to the spreadsheet if you're looking at the protocol. So basically the way this works, all the purple fields need to be filled out. The first field is how much media your thawed cells are suspended in. So usually that's just a mil. That's just when we warm them up and put them in media to wake them up out of liquid nitrogen. And then all the hemocytometer information. I'm not sure how universal hemocytometers are, but if they're the same, like standardized instrument, then this protocol will work for, Peter and Peter's lab, and that might be useful too. But yeah, you put in your cell counts here, so you do 4 cell counts, and then the dilution factor. That's what you can see. Dilution factor, how many mils your cells are reconstituted in, and your cell counts here. And then the green is just information it'll spit out. So that's your total cell count and your cells per microliter, which is pretty cool. But then here, once you tell it how many cells you want for each well and how much total media you want in the well, it'll tell you what to do for your master mix. And that's pretty cool. So how much of the thawed cells in 1 mil, or whatever the number is, and then how much other media there is. And then tells you how many how many wells you can plate as well. And then on the PBMC drug treatment tool, this is, I'm pretty proud of this one. I think this is pretty cool. It'll save a lot of time. This is, if you have a single drug dilution, you just put the information of the drug that you want to do, so the concentrated weight of the drug or the stock, and then what drug concentration you want to expose the cells to, and then how much media you swap out, and what your N is, how many wells you want to have treated. And then that'll tell you how to make the master mix. And I had it by default add 10% total volume for some pipette room, but I might take that out and just let you, increase the number here instead of having that be assumed. And then over to the right is if you're doing a dilution, so you have one drug and you want to do a bunch of different concentrations, you'd fill out the same information at the top, how much media you're swapping per well, the stock drug concentration, and then for every dilution you'd put how many wells at what concentration. So I programmed it for 5, and then all the master mix information would show up down here. And if you add another, let's say you want 22 wells, and we want to have that at 3 micrograms per mil, then that'll actually update the information of the first master mix that you make, because they all refer to each other. And if you want to make a second dilution, it'll tell you to dilute from the first master mix at the more concentrated dilution, and it'll tell you how much of that dilution to use and how much media. So I think that's pretty damn cool. And then that'll upload or update the numbers all the way up this little tree here, and hopefully save a lot of time. I think it'd probably save like an hour total of our time each time we're in the protocol. So that could be pretty cool. Anyways, hope it's helpful.